Hello, I'm Susan Wong. I'm a consultant physician at the University Hospital of Wales in Cardiff, UK, and Professor of Diabetes at the Cardiff University. I'm very pleased to have been part of this third International Diabetes Summit organised by the Celerum Diabetes Institute. And at this meeting, I have been speaking on the topic of gut microbiota. There's been a lot of interest in the gut microbiota, particularly over the last 10 to 15 years, where we have had increased ability to categorize and measure gut microbiota. In the past, we were only able to study those bacteria that we could culture. And over this last period, we have been able to sequence gut bacteria down to a very fine level, and both the sequencing to find out the classification as well as metagenomic sequencing has allowed us to make functional classification. So the gut microbiota, a very important part of our internal environment. The weight of the bacteria can be up to 1.5 kilograms, and the gene pool is more than 100 times that of the human genome. So they play a very significant role in our general being and metabolism. The gut microbiota have very important parts to play in the development of our immune system, and they will have effects on the adaptive immune system as well as the innate immune system within the gut. In addition to that, the bacterial metabolites are important for giving us vitamins, such as vitamin B12, and there are various other metabolites that include, for example, short-chain fatty acids, which can be absorbed for energy purposes and have other beneficial effects within the system. Bacteria are also important to produce lipopolysaccharide, which can induce inflammation in the system. And in terms of the topic that we are considering here at the diabetes conference, in terms of type 2 diabetes, it may be important that this lipopolysaccharide can generate cytokines and therefore have an effect on insulin resistance. There are also other endocrine effects where the bacteria may have influence on enteroendocrine cells, which lead to the production of GLP-1 as well as peptide YY, which can have an influence on appetite. So the composition of our gut bacteria is overall very important. There are many influences on the development of this bacterial composition. When we are born, the mode of delivery has an effect. Breastfed or bottle feeding plays a major role, as well as other influences in early childhood that include infections, taking of antibiotics, later on diet, and also something that is loosely called hygiene. So all of these components will set our gut bacteria, and this is usually by the age of 3 years. One very important feature of gut bacteria is what we call diversity. So thinking about type 2 diabetes, is there a particular composition? In fact, there is no general consensus about this, but what is very clear is that the medications for type 2 diabetes all have an effect on the composition of the gut bacteria. Particularly important is metformin, where the metabolites from the bacteria that are encouraged by metformin, that include the short-chain fatty acids, will be very important in helping to produce some of the beneficial effects of metformin. In addition, metformin encourages a particular bacterium called Akkermansia muciniphila, which helps to form the mucin layer. So when we think about gut microbiota, we need to bear in mind that there are various influences that will be set in the context of type 2 diabetes. So one might ask, could they be harnessed to give us beneficial effects? Could we use, for example, antibiotics, which is certainly not recommended because of other possibilities of detrimental effects of the antibiotics? Could we use probiotics? And at the moment, we don't yet know enough about the beneficial bacteria. And, for example, the Akkermansia mucinophilia can not be grown to a sufficient level to be used as a probiotic. Could we actually use molecules that come from beneficial bacteria? And this certainly is a possibility, but again, we would have to have manufacturing processes that could generate enough of this. But this is potentially something for the future. We know that bariatric surgery has, as part of the beneficial effect of the surgery in terms of correcting metabolic defects, some effects on the gut bacteria, and it will be important to understand more of this. And finally, for the future, perhaps there would be an importance for much more experimental techniques such as fecal transplantation. But at the moment, this is very much in the realms of experimentation. So at the end, I would summarize the importance of gut bacteria as being something that we should consider as having a major role in determining metabolism, immune system, and potentially something that is tractable for consideration of therapy. Thank you.